Okay, hello everyone. My name is Paul, and in this webinar series, I'd like to talk about the principle of holotomography system, as well as the application in biology and medicine. So, I'd like to start talking about the challenges in bioimaging, especially the optical microscopy system for the study of cells and tissues. So, as you know, the most of biological cells are transparent because there is just too little scattering. If you think about the compositions of biological cell, mostly proteins and some carbohydrate and mostly water and some the lipid and nucleic acid. So those are all transparent in the visible wavelengths. So that's why it's very difficult to directly visualize the individual cell using the bright field microscope, because in the bright field microscope, imaging contrast coming from the optical absorption, which doesn't provide enough information for biological cell. However, if you think about the human tissues, it composed of multiple layers of transparent cell. They look so opaque because of too much scattering. So the controlling this light scattering is a key for imaging individual biological cells and imaging through the turbid human tissues as well. And if I'm speaking of the histories of optical microscopy system, so as I mentioned, the biological cells are transparent. So even though after the development of the optical microscope, it was not easy to visualize the cell. So as you can see on the left-hand side, the cell looks transparent, but in order to solve this problem, the Dutch physicist, the Janneke, has developed a very nice idea, which is called the phase contrast microscopy system, and he got the Nobel Prize for that. So in the phase contrast microscope, the idea is convert optical phase information into intensity using interferometry. So essentially, by simply inserting the phase mask, you can convert the bright field microscope into the phase contrast microscopy system. So this phase contrast microscopy system is very nice for imaging live cell because it does not requires the labeling agents or the use of the fluorescence molecules and so on. But this is not quantitative. What I mean by that, on the right-hand side, even though you can nicely see the outlines of the cell as well as subcellular organelles, this does not provide the quantitative information about the thickness and height, volumes and surface areas and so on, or the even mass. So the people trying to develop the quantitative imaging system for the study of biological cells. So as you know, the scanning electron microscope or the atomic force microscope, for example, has been used for the study of the cell. They can provide very high spatial resolutions as well as the quantitative information, but those are not good for studying living cell because, for example, the electron microscope requires a metal coating to enhance the electron scattering for visualization, obviously. That kills the cell. So what you see here on the left-hand side is still shot of the dead cell. Right-hand side, you can see the AFM image of the biological cells, but they look, even though it provides two-dimensional information by scanning, which takes a huge time, however. So it is not a good idea for studying the dynamics of the living cell. So the idea of combining those two advantages, the quantitative imaging capability for the study of the living cell, has been the interest for in the field of biophotonics. So many people trying to address these issues, and one of the techniques to tackle address this problem is done by using the digital holography microscope or the quantitative phase imaging techniques. So the idea is very straightforward. All materials or any object consists of can be described by the refractive index distribution, which is intrinsic optical parameters determine the speed of light when the light passing through the specific materials. So on the figure in this slide, I have drawn the lead pearl cells schematics. And if you illuminate with the plane wave, because the refractive index of the cell, which is denoted as NC, is different from the refractive index of the medium, the Nm. So the light passing through the cell is slower than the light passing through the medium, which result in the formations of the wavefront distortion, so which is shown in the right-hand side. And since the image sensor, such as CCD or CMOS sensor, cannot detect the phase directly, what you measure using conventional imaging technique is just the amplitude information. So you cannot retrieve this phase information. However, if you employ interferometry or the holographic schemes, you can retrieve this phase delay information by the principle of the interference. If you introduce a reference arm, the sample beam interfere with the reference beam so that you can generate the holograms from which you can retrieve the field information. So this is typical images of two-dimensional hologram. Strictly speaking, this is a phase map retrieved from the hologram of the red blood cell. So because we are using the intrinsic optical parameter, refractive index, which will give a rise of phase delay of the sample, we do not require the exogenous labeling agents, so we can utilize the intrinsic optical parameters, refractive index, to visualize the cell in quantitative manner. So the height is retrieved in terms of the micrometer units, because you can quantitatively measure the optical phase delay. Using some approximation, you can retrieve the height information. So previously we have measured the series of the dynamics using two-dimensional the quantitative phase imaging techniques, such as you can see here, the white blood cell is crawling to the red blood cell, and after some interaction, the red blood cell is exploded. So if you increase the accretion speed, you can even do the further. For example, the red blood cell exhibit a tiny vibration because those are very soft and elastic biomaterials and biological cell, and they are showing the membrane vibration in the units of in order of 50 nanometers. And previously we retrieved the useful and mechanical parameters from these quantitative phase measurements. However, the old images I have shown previously was two-dimensional information. The two-dimensional hologram provides a two-dimensional phase delay map that does not provide the tomography information, only but topography information or the height information. In order to measure the three-dimensional tomogram system, you can get some idea. You can think about the principle in X-ray CT, computed tomogram system. So as you know, the X-ray CT can provide three-dimensional distributions of the X-ray absorption parameters of human body, so that you can nicely see the tissues or the organelle structures of the human bodies using the X-ray CT. The concept of X-ray CT is as follows. So sample, the object is a human body, and by scanning by changing the illumination angle, the directions of X-ray beam, and you can measure multiple two-dimensional the X-ray images, from which you can retrieve three-dimensional distributions or three-dimensional tomograms of the absorption parameters. And same principle can also be applied in the laser holographic microscopy system, because in this case, by scanning the illumination angle of the beam and measuring the multiple two-dimensional holograms, you can get some information about between the impulse and the response of this sample. And the objective here is to reconstruct the three-dimensional distributions of refractive index. So in fact, this is inverse problem. So this is not easy to solve directly, but using some appropriate assumptions, it is possible to retrieve the three-dimensional distribution of the refractive index. The first theoretical work was done by the Emil Wolf in 1969, and the first experimental demonstration has been shown in the 1979. But about that was about like the 30 to 40 years ago. But at the time, there are some scattered publication. But it was very recently that the many people has revived this field, and this concept of the measuring three-dimensional refractive index distribution becomes popular in the applications of biological sciences recently. So this is typical setup that we are doing these days. So this is called the field of optical difflection tomography system. So depends on the cases, depends on application. The people have come up with different schematics, but the main principle is the same. So using laser interferometry, the one beam, the sample beam, passing through or the reflected from the sample, and this transmitted beam from the sample is interfere with well-defined reference arm in order to retrieve the hologram information containing complex amplitude, which means the amplitude, real amplitude and phase information about the sample. So by changing the illumination angle, you can get different responses about the sample, and from those, the multiple two-dimensional information using the reconstruction algorithm, you can retrieve three-dimensional distribution of refractive index. So as you know, the refractive index can be described in complex number. The real parts correspond to the parameter determining the speed of light, which is the refractive index. And the imaginary part of the complex refractive index value correspond to how much light is absorbed. So by using the concept of optical difflection tomography system, you can retrieve the three-dimensional distributions of the absorptions as well as the light, path length delay. In the bottom, you can see rotating samples of the three-dimensional, the reconstructed refractive index tomograms of the red blood cell, and particularly for those red blood cells are infected by malaria parasites. So the left one is the early stage, and the middle one is the medium stage, and the right one was the late stage. Let me show you again. So those are the isosurface, which was the post-processed from the measured three-dimensional holograms. And since the parasite and cell body has different values for refractive index, you can nicely visualize the parasite, cell body, and as well as the reproduce the parasites as well. So for the past few years, our group and other groups in the world in the same field has demonstrated the concept of optical difflection tomography system is very useful for the study of the cell biology and the medicine because it has unique advantages. First, it does not require any exogenous label, such as the fluorescence proteins or the dyes, such quantum dots. It just utilizes the intrinsic optical parameter, that is the refractive index. So some cases it is very challenging and very inconvenient to employ the exogenous labeling process, but this concept of optical difflection tomography system is very useful for those sample, which is very especially for the case when you do not want to have the labeling the processes. Second one, it can provide quantitative imaging information because we are not measuring the qualitative information such as the light absorption, such as the fluorescence and so on. It can directly measure the refractive index, and that value, the value of the refractive index, very quantitative. So it does not depend on the setup. It does not depend on the sample preparation. If you have a specific sample, the refractive index should be the same values if you measure it correctly. So this is very reproducible, and it's very useful for the scientific studies. So there are other advantages, but I like to focus on the stress those two advantages: non-invasive capability as well as the quantitative imaging capability. So in order to develop the optical tomographic system, difflection optical tomography system, you need to control the illumination angle, or you need to rotate the sample without, if you do not want to change the illumination angle. So mostly previously people have utilized the galvanometric meters to steer the illumination angle of the beam impinging onto the sample, or people also utilize the sample rotation using the cylindrical the sample holders. However, those are not those, even though they have provides very the useful the demonstration, but this is not good for the commercialization. We realize because they have some limitation. For example, the 2-axis galvanometric meter is very expensive, and intrinsically it should have the mechanical instability, and the alignment of this galvanometric meter is very challenging. You need to do fine tuning every time, and the sample rotation is not a good idea for especially this soft and elastic sample, because the rotation of sample or the rotation of biological cells can change the shape and structures of the biological cell. So in order to solve these problems, by using this setup, these schemes, by using the mechanical rotation stage or the galvanometric meters or sample rotations, this is the one example, the setup that we have developed in our lab. And as you can see, the setup is very relatively complex, and the alignment is very complicated. And even though you have built up the system and working fine, after some time, or for example, once your lab matches hit the table, you need to spend few days to, well, a few hours at least, to fine tuning this system. So we realized this is not a good idea to spread, in order to spread the nice technique to the real world for those who are interested in using this system for the biological study or the medical applications. We thought maybe something new in order to control, a new technique should be developed in order to control the illumination angle. So we presented the concept of the optical difflection tomography system using a dynamic mirror device, which is DMD. So DMD is in fact the small, the MEMS-based electronic device, which is widely used in the DLP projectors. So it consists of the tens of thousands of the small micrometers, and each meters can be independently controlled. So even though this is the amplitude control, the amplitude modulator, if you cleverly utilize this concept, exploiting the concept of real holograms, you can utilize this DMD as the wavefront shaper. So we realized that this DMD can be used to control the illumination angle in optical difflection tomography system. And by doing that, we can achieve some, we can get some advantages. For example, this is very stable. There's not very moving parts, especially the large moving part is eliminated. Only small, the micrometer element is moving, which is very robust, which has been proved in the concept of the DLP projectors, and in addition, it's very cheap. The one unit is only the $100 for the price. So how we can control the illumination angle using DMD, and you can understand this concept can be understood by using this schematics of the light laser diffraction by the grating. So let's say you have projected the grating patterns, which is amplitude pattern of the DMD, and after the light, the reflected from the surface of the DMD, you will have the diffracted light. Some of the light will be transmitted, some of the light will be undifracted, but some light will be diffracted. But the angle of diffraction will be determined by the period of the grating. So by controlling the period of the grating, you can control the diffraction angle. So if you apply the well-defined intensity patterns on the grating of the DMD, you can utilize this DMD unit as similar to the galvanometric meters as done in the previous old optical difflection tomography system. So using this concept and also optimize the other optical the geometries and integrations, we have come up with this standalone device that will be on the market in 16th of February at Photonic West. So light source, the image sensor, the DMD, the object lens, everything is integrated. So all you need to do is just insert your sample, the sample holders, and everything is automated, and which also generates three-dimensional reconstructed images by using this our softwares. So as you can see, the XY, XZ, and YZ cross-sectional tomogram, the reflecting tomograms, as well as you can also provide some the colors on the reflective index values for the better visualization. And this is typical images that we have measured using the setup. So the top row is a lead blood cell, and as you can see, you can nicely capture the donut shape, the lead blood cell, and the middle one is small lymphocytes, and you can visualize the nucleus and the cell membrane. So by putting the artificial color renderings to the measured, the reflective index values, we can nicely visualize those three-dimensional shape, three-dimensional tomograms of the reflective index, and which is very similar to the conventional, colorimetric images are very comparable to conventional, H&E images or the confocal images. The bottom one is hepatocytes, and you can see the nucleus and subcellular organelles. And we can also measure the 4D, the reflective index tomogram. So you can measure time-lapse images of the three-dimensional reflective index distribution. So by measuring those movies, you can investigate unprecedented the images, some very novel images that was not possible using the previous conventional optical microscope the scheme. So, and I like to present some of the representative data, the sample that we have obtained using the setup. So this is lead blood cell, and you can nicely see the donut shape. And this is the macrophages, one of the types of the white blood cell, and you can see the subcellular structures as well as the membrane of the cell. And this is the hepatocyte, the cell in the liver, and you can see very nicely this lipid drumlet having high reflective index, and this is one cell, and this is cell membrane, and this is nucleus membrane, and this is nuclear light. So without using any exogenous labels, the measurement of the three-dimensional reflective index tomogram can directly visualize the subcellular structures of the living cell. And this is one example of 4D. So this is HeLa cell, which was attached to the glass substrate, and by adding the trypsin, those cells are attached from the substrate. So you can see that the cell becomes like spherized, and it will just pops up. And we also measured the apoptosis, programmed the cell death, and the hallmark of apoptosis is blobbing formation, and you can see that the blobbing, and after some time the cells stop moving like this. So by measuring the cells, we also observe very interesting the blobbing formation, which is very large compared to the previous one. And we can also visualize the other subcellular organelles, such as the nucleus and so on. And this is one example of the cell undergoing the necrosis. Please watch. After some time, boom. So this is like the cell die. Okay. So for further information, I'd like to introduce the very nice book written by Gabriel Popescu, and also you may want to refer to the condensed review paper from our group. And there are the conference series hosted in the Photonics West. So first conference was held in 2015, and the 2016 we have second conference, and the number of people and number of groups are increasing for those who are interested in the topics of the this emerging field of quantitative phase imaging. So for these talks, I have briefly mentioned the principle of the holotomography system or the optical diffraction tomography system, which enables to measure three-dimensional distribution of refractive index of the biological samples that can provide the unique advantages compared to the existing imaging techniques. Okay, so thank you for listening, and if you have any questions, please contact me by emails, and I'll be very happy to answer to your question. Thank you.